Hello and welcome to the very first episode of a new SciPod, a platform where we bring research and knowledge in biology, chemistry, and medicine from experts to you. Whether you're a student, a researcher, or simply someone with curiosity about the world, this podcast is designed to make complex ideas accessible and interesting to you. So, my name is Ambek Bekturgan. I'm first-year master's student majoring in life sciences, and here is Alisher.
I'm Alisher Mustafayev. I'm second-year medical student in Nazarbayev University School of Medicine. For today's episode, we're thrilled to have Associate Professor Damira Kanayeva from Biological Sciences, who joined NU in 2011. Since then, she has been actively involved both in teaching and research. Dr. Kanayeva established research direction in the field of biosensors and biodetectors at NU, which represents a promising option for developing reliable, simple, and affordable tools for the effective detection of disease-causing organisms. Apart from research expertise, Dr. Kanayeva is involved in supporting and mentoring students' clubs, including us. We are very grateful to have the professor as our supporting mentor.
So, today's conversation with Dr. Kanayeva promises to take us deep into the topic of the tuberculosis disease, covering everything from fundamental mechanisms to their real-world applications. So, sit back and join us as we explore the frontiers of science, one conversation at a time.
Nice to meet you, Professor.
Yes,
we are
Professor.
very pleased to have you as the first guest. How are you feeling today?
Thank you so much for your nice words and introduction. I'm really happy and honored to be here and we'll share maybe some knowledge and experience with you and your audience.
Well, that's great. Thank you so much for agreeing to be our mentor in this project. It really means a lot to us. We really appreciate your guidance and expertise, and we're of course curious, what inspired you to take on this mentorship role with us? What is your motivation?
Well, it is quite interesting, right? I work on daily basis with students and I see motivation and this is actually one of the first scientific podcasts, right, at Nazarbayev University that my students shared with me, and I see the enthusiasm and I see that they really want to contribute to the field of not only the science but also disseminating science and knowledge, right, to wider audience. So I think it's as being a faculty, it's our not only job but actually I see it inspiring to support our students so that they can also contribute to this field as well. So yeah.
Wow, that's great. Apart from being a professor and taking part in students' life as in ours, you are also a mom. How does it feel to be both a mom and a researcher? Is it hard? Probably.
Well, when back in 2011 when I joined the university, right, I was just returned to Kazakhstan from my PhD studies. Back then I was single, then I got married, established a family with my husband. And of course, it is quite challenging, right, to keep up research, science, teaching, right, mentoring students, graduate and both undergraduate, and having kids as well, right? So it was a new experience for me, a challenging one, but having supportive family, students, colleagues, right? It actually means a lot and actually helps a lot, right? To grow as a, not only as a person, as a mom, as a scientist, as a faculty, right? So of course it requires commitment from both sides, but it also requires support, tremendous support from the close people. So I'm really thankful to my students, to my mentors. I had also mentors back then, Dr. Dr. Baygarin, who I really closely worked with. So it actually meant a lot and it actually eased up my load. And my family, of course, husband, because when I'm here, right, someone also needs to take them to the school, right? So having a supportive spouse is quite important. And obviously the grandparents, right? Because grandparents in our community, they play a big role because they also quite, not only in terms of the education purposes, but also showing the family merit, right? Is quite important, and they helped us a lot.
It is actually quite complicated to to be both an academic professor as well as a parent, as
Right.
a family member. So, do you think your profession actually affects your approach to parenting?
Well, since we are biologists, right?
Yeah.
So we understand more or less better in terms of like the kids' development, the infection that they may catch, right? The importance of the vaccines in timely manner is quite important, right? So in that aspect, it obviously helps me a lot. But of course, as it doesn't mean that I can predict something, right? In terms of the behavior or something. But in terms of the development, yes. But usually parents, they should be connected. They should be in touch with their kids so that they do not miss certain, like, I don't know, milestones, right? Because, like, for example, I have two kids, daughter and son. Daughter is 11 years old. My son is 8 years old. My daughter is approaching this teenagerhood, right? So no one would be prepared and no one can actually be ready. But I'm trying to see from the other perspective. So from the science perspective as well, and obviously sharing, like I also listen to different kinds of podcasts in terms of the kids' development, so it helps a lot. But in terms of the infectious diseases, development, yeah, from that background, I guess having this background and education helps
Yes,
a lot.
this is definitely helpful.
It's like usually people who is like going in academic sphere doing researchers, they are more their thoughts, I guess they are thinking more in a structured way. So like it is expected like the kids of academic people are usually very structured, living in kind of strict conditions, but we can see like that it is not like that always. It really depends on the person itself, basically.
Yeah, but also nowadays we can see that even the societies, right, they are moving towards like early development of the kids, right? So, for example, back in 2013 when my daughter was born, so I was really like eager so that her development, because she was preterm, I had a preterm delivery, right, on 36th week. So that she would catch up in terms of the development, right, with the ones that actually who are her same age. So I was like, I took her to the swimming lessons back then when she was only 8 months old. So nowadays, what I see is that she's actually doing her. She's really into swimming lessons. She's actually participating in artistic or synchronized swimming. So, and I think nowadays community is more or less ready, right? And they understand like what we need to actually give our kids, right? And there are different kinds of clubs out there, right? The Montessori or early development. So I think it's quite good to see. So I'm just sharing my personal experiences. Yeah.
Yeah, so basically you give your daughter a new hobby with the artistic swimming. That's fascinating, yeah, for sure.
But also, I think I always think like when I get older, right? And I'm not here. If, for example, I'm not here with my kids, right? So they need to have the skills, right? The more skills they have, the better. For example, myself, I cannot swim. So it's like my kids, they should learn how to swim, and like if something happens, they have this skill developed, right? It's like kind of a driving, right? If we can drive, we have this driving kind of, what do you call it? Not driving lessons, but license.
Yeah.
So obviously, you are more mobile, right? You can get around, you can travel to far distances, right? So that's kind of a skill that if we have more skills accrued from the early childhoods, then I think we're better adaptable, and that's what I want to see in my kids as well.
You went through a long journey to become an associate professor at Nazarbayev University. We are curious about what initially drew you to this field and what has motivated you to pursue microbiology specifically.
Yeah, that's a very interesting question. Thank you. Well, I graduated from high school. That was the late 90s. So maybe your parents may recall that was quite challenging and tough times for our country. So back then, science was not popular, biology was not popular. I mean, like in terms of like getting a diploma, right, in these fields. because most of the scientists back then, and my parents, for example, they are scientists, right? And they are in academia, in biology, and as well as in chemistry fields. And they greatly impacted, I guess, my choice as well. But that was not actually something you would, in terms of the financial perspectives, benefit, right? So, but you have to have motivation, and you need to see from a bigger picture, right? So what actually would be a driving force, right? And nowadays we see that how this kind of changes, right? Kazakhstan went through, not only in terms of the financial, right, and the country standing, but also in terms of that nowadays, like most of the people, they understand that the driving force of the economy is not only like oil or gas, right, but also science, right? If we do not have science, if we do not have scientists contributing to this field, then how we're going to educate our kids, right? How we're going to proceed in terms of the science and development, right? So that was kind of, I believe back then, I was back then only 16 years old, so that was the motivation for me. I did my internships not only in Al-Farabi, Kazakh National University, that's my alma mater from where I graduated, got my bachelor's degree, then master's degree. I also did internship at the Institute of Microbiology and Virology. So I saw from different angles how actually the science in Kazakhstan from the late 90s and up to the 2024, it's been like almost 25 years, right? So we can see there's a big improvement, right? Not only in science, but also in financing the science, right? So that we get more bright minds, young people, right, joining and actually contributing to this field. And that's actually something rewarding to see. So then I went to University of Arkansas in the US, and I saw from a different angle how at an international level, right, the science is conducted back in 2005. So that, I guess, was the motivation. So I was like, biology is not only about biology, right? It's biology is integrated to our lives. So it is a multidisciplinary field from chemistry perspective, from medicine, right? Because it is a translation, right, of research from the bench to the patients, right, to clinics, to diagnostic laboratories. So that was my motivation. And infections, like microbiology, like pathogens specifically, right, we need to understand what actually induces this infection, right, how to actually correctly diagnose, right, how to actually make those diagnostic tools as less expensive as possible and sensitive on the other side, right, and specific, right, not compromising these two factors because they're quite important. So I don't know whether I have answered the question or not, but that was kind of the picture I had back then. But now I see that I made a right choice. We should understand that while choosing a profession, we shouldn't be only motivated by the money that we make, right? In that, but what contribution we make, right? In the end, if we're contributing, that would be rewarding, right? And I think money will come in as well, right? If there is a contribution.
Yeah, sure.
Yes, it's always actually a spark inside person that should drive people, not the money, not the material stuff that we could get out of our jobs, but the inner drive force. So you have mentioned that biology is actually multidisciplinary. We have a lot of fields and integration with chemistry and mathematics and the medicine as well, but you decided to choose the microbiology. So I wanted to ask, what fascinates you about microbiology the most?
Well, microbiology is about microorganisms, right? The living entities that we do not see. So all of us, we were in pandemics. So we have been exposed, right? Or were exposed, I hope, right, to those pathogens, specifically the SARS-CoV-2 inducing this COVID-19. So nowadays we see the huge impact, right, of those tiny items that we do not even see in nanometer scale, right, the pathogens. But they have a huge impact, right, on our well-being, right, because those infections, the pathogens, they communicable disease, right, and they could be airborne, they could be foodborne, right, they could be waterborne infections. So because of that, right, we should study these fields.
Yes, definitely. Actually, I think most people don't have like second thoughts about microbiology? Like why would you study like things that we don't see with our eyes, etc.?
Yeah, but it's not only about the also negative impact, right? Even like to build the immunity, right? We need to have a good microflora, a good microbiome, right, residing in our body surfaces. And the more good microflora we have in our GI tract, then the better we are, right? So that we will not be susceptible to those pathogens, right, which we are exposed to constantly on a daily basis, like every single minute, right? There are airborne pathogens that can actually circulate with the air droplets, right, or the particles. So not allowing them to colonize the body surfaces is by having the good microflora, right? So, and having probiotics, I don't know, maybe as supplements or fermented food samples, right? So that could be also how to build this good microflora.
Yeah, and now elaborating more on your background, you were assisting research at Al-Farabi University in Almaty and Arkansas in USA. You were doing PhD program in USA and conducting research at the NU now from 2011 up to now. Do you have any observations that differ in these three institutions? Well, elaborating more on observations is like the different nature and locations of these universities, maybe the mentality of the places, how it is different, and of course, probably for our dear listeners, the things will be like very interesting. For our like undergraduate students, is like internships, and for researchers, is of course the fundings for the research, how it differs in places that you have worked.
Thank you so much for this question. So I worked in different years, right, in these three institutions. So as an undergraduate student and also as a master's student, I was in Kazakh National Al-Farabi University. So I'm always thankful to this university, to the Department of Microbiology that actually gave me this foundational knowledge, right, to build my experience, skills, right, and build upon my career. So I would be always thankful. But back then, I cannot compare, right? Back then, in the late 90s and early 20s, I cannot compare to the nowadays, because nowadays Kazakhstan is actually investing a lot of money, right, into research and education, compared to back then, the late 90s, because we had different things, different problems, right, those days. So, but nevertheless, right, the university used to be as one of the leading universities, not only in education, but as well as in science. And biology department over there was established as one of the departments, the first departments back in 1934. So it's more or less like 100 years of history, right, of this university. So, but they have these foundational departments in microbiology, in plant science, right, the zoology. But if we're comparing, the next university that I worked is the University of Arkansas, right? I worked there between 2005 and 2010. So in this university, because it is located in the central part of the U.S., right, and they are more like agricultural university. They have the investors who are sponsoring their research for the agricultural purposes. So let's say for poultry, right, the chicken. So we think like, oh, it's just a poultry, right? What to study over there? But they would study the immunity, right? The infections that are associated with this animal, right? Domesticated animal. How to diagnose and detect the infections or the pathogens that can actually be found in these poultry samples, right? So, like within several minutes, so that the yield of this food sample would be higher, right? So that the consumers would get a safer food, not contaminated with pathogens, right? So they would look at it from different perspectives. They would have these plant science departments. They also have, very interesting, I didn't know this before I joined the university, that they have the program specifically in business school. So, for example, all of us know Walmart, right? It is well-known, not only grocery store, right? It is a supermarket, right? Where all the Americans, nowadays even in China, they have their stores of the, they can find everything, starting out from bread, milk, and tires. One can go and shop for tires, for example, yeah? The headquarters of Walmart is located in Arkansas, right close to the university I used to study. So they would have this business school, right? So Tyson, for example, that's, I'm sorry if I'm calling these names without the commercial brands, but these are the kind of driving forces, not only economy, but as well as the contribute greatly to the science and research development in this particular region, which is quite fascinating. And I think we also, in our country, we need to see like what particular regions, right, have like big companies, and they should have specific problems, right, that should be tackled through science and research, and potentially they could contribute to this field and help out those regional universities, right, where those are located. At Nazarbayev University now, it's in between 2011 and 2024 up to today's days, right? So, well, when I joined the university back then, the university just was recently opened, so we didn't have the wet labs. So we had to actually create those labs, right? Because we participated in retrofitting the facilities. There was no C4 building, research building, right? So we participated in designing, establishing new laboratories. So in our university, we are more of like, and you guys are know the faculty members, right? In our department, biology department. The concentration it's also multidisciplinary, even though we are not quite big department, right? But the field of the research is quite multidisciplinary. For example, in my lab, we do biosensors development, right, which is quite complex and multidisciplinary, involving the knowledge not only in biology, microbiology, but as well as in chemistry, analytical chemistry, plus, for example, engineering components, right? So it is quite challenging, of course, from one side, because we need to prepare and introduce students, right, to these fields. We have faculty members who are in microscopy, right, atomic force microscopy, in immunology, right, in cancer. So even though we are a quite young university, but we made, I think, our also quite big impact in terms of the research and development in the field of the biology and multidisciplinary fields. So just roughly to kind of describe.
So back then, when you joined the Nazarbayev University, there were no labs. And when the university decided to build the labs, they were asking advice from you, if I understand it correctly. Like, what equipment should be used for different techniques, different research methods? What is like?
Yeah, so back then we were only a few people working in the department. My colleagues and I, so we were kind of not only drawing the wet labs, right, but trying to equip and purchasing all those equipment, right, so that the students would have this access to the equipment and facilities, right, as in most of the international laboratories, universities. So, and let me share this story. So when I joined, so I established my lab, right? So I was working on a project that I just started in 2011, and late 2011, I asked my former supervisor, Professor Dr. Yambin Lee, to come and visit the university. So he came from the US, from the University of Arkansas. That was quite cold back then in December, -30. So he came here, and he was surprised and astonished, because he's like, Damira, we like in the US, we don't have this brand new laboratories, right? Because all the buildings, they're quite old, they maintain them right well, but most of the equipment, they also have been aged, right? Because to get the funding and to purchase the equipment is also quite expensive. So he was quite surprised, and it's like, you guys have all the facilities, equipment, and the funding and support, right, from the not only from the university, but also from the government, to educate and educate students and establish this laboratory and conduct this kind of high-end research. So, yeah.
So we're going to move on the next session. On the next section, well, it's mostly about your research, elaborating more on scientific topics. You have a research focus on the detection of different diseases such as tuberculosis, COVID-19, and cancer. In this podcast, we would like to focus on tuberculosis more, taking into account its global relevance. On the most recent dates, I guess, on the most recent data by World Health Organization, I guess Kazakhstan was placed on 12th place by spread of tuberculosis, by rates of tuberculosis disease, which is resistant to drugs. Well, I read it from the internet. Most of us have heard of tuberculosis, of course, but could you briefly explain what it is and how it affects the body?
All right, thank you for this question. Yeah, so unfortunately, yes. So again, going back to 2011. So when I came in and joined the university, so I was trying to see like what kind of problems we can tackle here in Kazakhstan, right? So obviously in the US it's a little bit different picture, right? But I cannot just go ahead and do the same things as I used to do in the US. So in the US I used to study Listeria monocytogenes, which is a foodborne pathogen. But over here, like we have another problems, right? And tuberculosis is among them because I looked at the statistics, WHO information, right? The official numbers, right? Because if unofficially, there are also patients, right, who suffer from tuberculosis, but they wouldn't actually register and do not get the treatment, right? And spread the infection. So tuberculosis, what it is? Unfortunately, we are on the list of WHO, even though WHO is actually has this aim to eradicate this infection, this pathogen, right, by 2030, because most of the countries, right, they do get vaccines, and vaccine shots they administer to infants during their first days, and then to be reimmunized at the age of 6. So it is a intracellular pathogen. So, which is considered more gram-positive, gram-negative. So it is more of like microbiological terminologies that is based on the cell wall characteristics. So nowadays the challenge is that it's not because we cannot contain these infections. We can, but there are preventative means also developed a long time ago, right? That's vaccine shot, the BCG vaccine shot. But unfortunately, there are statistics that because of certain diseases, right, for example, the malnutrition, not having access to diagnostic tools, right, or the patient, they just afraid of going and seeing the doctors, right, and getting correct diagnostics, because if not correctly diagnosed, then there's no treatment, right, for it. And the biggest challenge nowadays is that it is the resistance, and resistance not only to one single drug, right, to multi-drugs, multi-drug resistant strains of tuberculosis. So that's a challenge for the community and for doctors while treating the patients. And obviously there are also co-infections, right? So there is a high relationship of patients who have like smoking habits, right, who also have those HIV or the AIDS, right, infections. They are highly susceptible to these infections, and not only tuberculosis but also to opportunistic infections. Opportunistic means that those microorganisms that we have in our body surfaces, they may turn against us because the immunity has been compromised and the numbers may increase, right? And like we talked with my students the other day about the Pseudomonas aeruginosa. It is one of the also opportunistic pathogens that may actually flourish when immunity is compromised. So unfortunately, yes, in Kazakhstan that is still a problem. So we're trying to also contribute to this field through developing like diagnostic tools, biosensors, aptamers for the rapid detection and identification of this pathogen.
So that was a very interesting answer for our question. Thank you very much. We know that tuberculosis was an issue ever since back in the 18th, 19th centuries, and back then the mortality rates were really high, like around 70% of all infected people eventually die out of tuberculosis, and currently the statistic is far better. Like it's still not great. It's around like 50% people still die if they get infected, and worldwide we still get around 10.6 million people yearly get diagnosed with tuberculosis, and around 1/10, like 1.3, 1.6 million people die eventually out of tuberculosis. So I'd like to ask, why is this disease still an issue since around the world, even though it was discovered back then?
Yeah, so this pathogen, right, it has been around for many years, for centuries. So it seems like we have the preventative measures, the treatment, antimicrobial drugs, right, to treat the patients. But this, as I mentioned earlier, the drug resistance, right, is the key issue. But also, when we do this annual screening, right, of chest X-ray, or so we get also screened, right, for the specific pulmonary infections. So, however, when it comes to the infection, it's about the sensitivity, right, of the diagnostic tool, and as well as the specificity. So the earlier we know that there is an infection occurring, right, the sooner the treatment will take place, and obviously the outcomes will be better. But the X-ray is not quite sensitive. What kind of techniques, first of all, are used, right, in diagnostics? These are bacteriological culturing, and this microorganism is one of the slow-growing microorganisms. So if to culture on the traditional media, then one would be observing the growth of the colonies of this microorganism only up to 50, 60, almost 2 months, right? So which is quite long, right? Obviously nowadays we have new techniques, rapid techniques developed. One of them is PCR, gene expert. So this technique allows within one working day, right, to get the information whether the patient is having, let's say, this infection, the tuberculosis, and whether there are MDR also strains, right? So it can discriminate. It is quite sensitive, right? But it is quite also costly, because one cartridge is used per patient. And the other technique, which is quite yes or no, it's not about the quantity, right? It cannot quantify, but one would be donating, a patient would be donating a sputum, and then one would be actually, the technician is to go and look at the slides of the under the microscope, right? And do the staining. Because it is nor gram positive nor either gram negative, we cannot use readily this gram technique staining. The technicians, they would use the acid-fast staining in order to penetrate through this mucous mycolic sheaths of the sputum layer, right, and also the this mycolic sheaths that would be coating the bacteria. So in such cases, it's about the skills of the technicians, right? How well they would be preparing the sample, and whether they would be able to discriminate other microbes, right, from that of the Mycobacterium tuberculosis. So it's about sensitivity and as well as the specificity, right? So and also it is an airborne pathogen, right? So in densely placed communities, patients, they may trap this infection easily. So I'm not saying Kazakhstan, we are densely packed, right? But it's still in one single household, right? Usually, in families, we have extended families, right? Kids, parents, then grandparents, right? And obviously, we share the more or less same, not same, but the common microflora among this household community. So that could be also one of the issues. Co-infections, right, could be also an issue, and multi-drug resistance, because this treatment of the tuberculosis infection is quite lengthy. It's not about 2 weeks only, right? The treatment is up to 6 months with up to 5 antimicrobial drugs. So it is quite harsh treatment, I would say, which may lead to certain side effects in patients, and yeah, so it is still a challenge in the community.
So it's if you're getting tuberculosis, you're probably going to be out of your work, out of your life, basically for 5-6 months. As Dr. Kanayeva mentioned, yeah, it's a very hard disease to be also cured from, so please stay aware. Especially it is airborne, tuberculosis it is caused by bacteria that is mostly given by airborne way, so please stay safe.
One more thing to add, and also latent, right? Because the patient may have this infection, but they are not showing the symptoms.
Yeah.
So there are dormant cells, right, that could be also observed. And as a result, because the patient is not showing the symptoms, right, and the cells are not actively dividing, but once there is a advantage, right, they would seize it, and they may actually awake from the dormancy, right, and as a result, there would be flourishing of multi-drug resistant strains of this microbiome, so which is quite challenging to treat.
So when people get infected with tuberculosis and they face a lot of complications, health complications, like for instance in later stages I think it's joint and lung failures, as well as a lot of problems with liver and kidney. So even though we have currently technological and medical advances, treatment of the disease still remains like a difficulty, I would say, for us. So what would be the main reason for such an issue in the treatment?
The treatment. So yeah, I think you've answered your answer, because the side effects, right, not after, even while getting the treatment, right, is usually an issue for the doctors, because they would see that the patient is not coping with the dose that they are getting, right, in order to inactivate those microbes. And side effects, and usually while there are statistics in terms of the morbidity, the deaths of the patients, right, it's not about specifically, right, about the tuberculosis itself. It could be also associated with the side effects, right, because the failure of the body, right, the organs to actually get all those drugs, right, pumping in to inactivate the microorganisms. So the statistics, I would say, it's not only about the tuberculosis, but also about the side effects, right, of the drugs and the effect of the
so we can kind of logically connect it, because why there is problems with kidney, for example, or when taking the drugs, when taking the treatment from the tuberculosis, because of course the side effects are usually affecting from heavy treatment. It affects other organs as well, especially our filtering organs. It's on kidneys, so it's quite logical, I would say.
Yes, sounds logical to me.
The difficulties that we mentioned can be also the result of late diagnosis. Well, for a lot of medical cases, the late diagnosis is the case why there's a high lethal mortality rates as well. So to prevent such complications, early detection of diseases is essential. So basically, quite big part of science is working on that sphere. So we can really see that speed is a key for in-time treatment. Let's then discuss about diagnostics. What diagnostic methods do you have nowadays, and what's coming new to our science society?
So I briefly mentioned some of them. So microscopy is one acid-fast staining microscopy technique, right? Then is the PCR using the gene expert usually. That's what most of the TB dispensers would use. And then the next step is, for example, there are different kinds of techniques, also rapid techniques developed, right, in terms of the antigen sensing, right? It's lateral flow, or there are also techniques called as IGRA, so they would detect the also specific cytokines, right, which are secreted while there's an infection occurring, right? The other week I have attended the biosensors workshop where my colleagues, they were sharing also their findings, and I was asking them in terms of the specificity of the SIGRA, right? It is the technique itself. It is quite specific in terms of the detecting the infection, but it is not known what specific infection, right? So they say they would get the samples from pleural fluid, right? So that it is only related to tuberculosis. But I was like, well, nowadays we also observe with SARS-CoV-2, right, because it can also colonize the lower pulmonary tract, right, and induce the infection, viral infection, compared to tuberculosis, which is a bacterial origin, right? So could it be also complication, right, in terms of the correct diagnosis of tuberculosis versus the SARS-CoV-2? So it's something also to address. But for example, in my lab, what we do is that we work with biomarkers which are secreted in the surface of the Mycobacterium tuberculosis, and it is called as MPT64 antigen, and it is secreted only in actively dividing cells. why I'm saying this, I'm stressing on the actively dividing cells because, as I said, that there could be latent, right, or dormant, right, or there could be, for example, after the treatment, there could be also circulation of the pathogen, right, in the body fluids. That could be sputum, that could be urine sample or blood sample, right. So we only want to detect actively dividing tuberculosis bacteria cells while detecting the specific biomarker secreted by this pathogen. So that's called biosensors. So shall I move on to this field?
Yeah. So we are actually heading towards that. So in your research lab, you mostly focus on early detection of such kind of diseases using the biosensors and especially aptamers. So could you please give our explanation to our viewers? What are these biosensors and how could they be used for diagnostic purposes?
Right. So as we're advancing in science and technology, so we are not studying in one place. So this biosensor field, right? It is bio and sensory, biological sensor, right? So we're detecting biological substances in this case. So it contains this the transducer, right? That has the specific receptors functionalized over the surface. When it comes to receptors, we may use different substances. For example, antibodies, right? Or let's say enzymes, right? And upon binding to their target, for example, in our case, we use aptamers or antibodies. They will change the conformation and they bind to the antigen. So while it binds, it produces a specific signal that we measure the change, right, compared to that of the control. So in my laboratory, my research team and I, we have selected a pool of aptamer candidates against MPT64 secreted protein of Mycobacterium tuberculosis. What are aptamers? Let's first answer this question. Yeah? So the aptamers could be RNA-based or single-stranded DNA-based molecules. So everything comes from nature, right? In nature, we also see this kind of nucleotides, right? Non-translating, non-coding nucleotides. They're called as riboswitches. So, like microbiology 101.
Well,
And they change the conformation of the sequence upon binding to the antigen. So we try to also, we are not the only lab working in this field, there are a number of labs, but our contribution is that we have selected specific and sensitive aptamers, the single-strand DNA strands, working as alternative to antibodies. So they have high sensitivity and specificity, so we conducted a number of experiments, not only in the laboratory setting, we have also tested in clinical samples, we have analyzed sputum and as well as serum samples to observe the sensitivity and the specificity of the aptamers that we have selected. So we combine these two technologies, the aptamers and as well as the biosensor, the electrochemical sensor, and then trying to develop this rapid detection tool that allows to screen the samples, right, and effectively, sensorily, specifically detect the target pathogen.
So like aptamers is a new method as we are like understanding it. So do you think that the future diagnostic methods that aptamer will replace now methods that are like widely used now? Do you think the future, basically, do you think that future is behind the aptamers?
That's an interesting question. Well, so we are competing. So we all need to understand, right, who are the competitors of aptamers.
Yeah.
Those are antibodies, right? And we're saying like we can use aptamers versus antibodies that do not require, while selecting and synthesizing, do not require animals. Versus antibodies, we need to use animals, right, to get those antibodies specific for these targets. They also have a longer shelf life, right? So because they are single-stranded DNA, right? Upon knowing the sequence, we can just synthesize them. We can add different tags, modify them, right? So we can play around with the sequences, so that which our antibodies may not have. But also, while talking about the competitors, so the industry in terms of the antibody is quite huge. We have a really big players, right, in the industry. There are big companies, right, that would sell and produce antibodies. So it's about the competition also, right?
As well, yeah.
Yeah.
So entering into the market for the aptamers, maybe at one day, right, it would be, but at the time, it mostly at the research scale. However, I need to mention there are also aptamers that have been actually patented and they have been released as therapeutic agents. So they can be used not only as diagnostic tools but also as therapeutic agents. So if you Google up there, you'll find a number of aptamers that have been actually FDA approved, Food and Drug Administration in the US, as a treatment for the patients. But there are advantages certainly that we need to, I guess, work on so that we can also be quite at the same market as the antibodies.
So we are leading to that question as well. I really hope that one day Kazakhstan will be placed on the world map as a major antibody and probably aptamers producer. Well, I guess we'll together cope for that for the sake of our country. What makes aptamers special compared to traditional ways of diagnosing tuberculosis? And why are they potentially better? So we are coming more on the advantages of the aptamers.
Yeah. So what we do is that so we develop the sensors which are when we use aptamers and functionalize the surface of the electrodes. So when the if we want to, for example, manufacture, right, and make it as a mass market or put it into the industry, so the shelf life, right, plays a crucial role. So we're in Kazakhstan, and all of us understand, like, and my colleagues would understand that whatever we receive, like in terms of the reagents, proteins, antibodies, it takes quite a huge amount of time and effort to bring them in here, right? So in such cases, one cannot actually guarantee that the samples will be maintained at a certain temperature as the manufacturer actually put on the label, right? It's I don't know, plus 4 degrees Celsius for this amount of time, right? So in most cases, they may, by the time they get here to our labs, they may lose their efficacy, right? So the antibodies, they are partially working, or we have this shorter shelf life, right, to work with only 1 month. But then obviously we are not throwing them away, we are trying to make use of it. But again, it's actually something that would be concerning us. So in such cases, aptamers would be a better choice because they can be transported at a room temperature because they are single-strand DNA or RNA sequences, right? They are not susceptible to temperature fluctuations. We can modify them if we functionalize the surface of the electrodes so that they would be ready to be used, let's say, for the diagnostic purposes. They will not be degrade as fast as, for example, antibodies would do, right? They would be quite sensitive towards them. So in these cases, it is quite important for us to have this kind of recognition elements, aptamers, that tolerate these conditions.
So when we were talking about like developing the reliable biosensors, you said you talked mostly about the aptamers. Would you say that the aptamers is like the most challenging thing in development of reliable biosensors, or is it something else that could be also part of it?
Well, in terms of the recognition, we use aptamers, right? But other colleagues, for example, in other universities, they use different recognition elements. They may use antibodies. For certain projects, we also use antibodies, right? Some research laboratories, they selectively, they synthesize this MIPS. They are molecularly imprinted polymers as recognition element, right? Chemist people. So the application, or I guess as we, like scientists, we are creative, right? We try to bring our knowledge into this field. But for us, in this setting, because I'm a biologist, right? For me, because I'm familiar with PCR techniques, right? Because it requires this PCR techniques for the evolutionary aspects of the sequences, right? Using the Celix technology for aptamers is something that I'm doing. Getting the sequences and then applying them and testing for the characterization purposes, but also for the detection and identification purposes as well.
Well, now that you have mentioned like other also techniques that applied for like biosensor detection, how do you see the future of tuberculosis detection for like in the next 10 or 20 years? Could it like someday we'll have at-home tests for tuberculosis that everyone could use, that will be accessible?
That's where we are aiming at, actually, right? Because the faster we know the results, the better choices we make, right? The better, the sooner we would get the treatment. I mean, we, the patients, right? So let me give you this example. So most of us, right, in our homes we have this glucose monitoring equipment, right, which is quite small, portable nowadays, right? But back then, in the previous century, in the 20th century, it was a benchtop instrument. So nowadays we make it as portable, right? So that we can, from a finger prick, we get the blood sample and test the level of the glucose, right? So it is at home measuring equipment and technology. So nowadays, if you take a look at the scientific papers, there are, for example, skin patch tests, right, that allows to detect the level of different body fluid biomarkers, even the pH, right, or lactate, for example, for sports people. So in our case, what we want to do also make it this device that we are developing and studying, right, to make it accessible to wider community, so that for example, in the rural areas where these X-ray machines, right, or the bacteriological laboratories or the PCR instruments are not really available, we can bring in this kind of electrodes, right, the instruments, so that we can test them on site, right, from patients. Let's say we have a sputum sample or urine sample, right, and whether they should be sensitive, right? The sensitivity and specificity is the key in every diagnostic tool that are currently available, including ELISA, for example. ELISA has been around for a number of years as well, right? And biosensors field is also, if you take a look into the history, so it was evolved actually, if I'm not mistaken, back in 1980s. So it has been building up, right, for the past 40 years, and we can see the market is actually growing. So even this watch, right, the commercial that we are wearing, the watches, right, they monitor our heartbeat, right? They monitor our the level of the ECG, right? What else? We can put the skin patch, right? There are lenses also that would actually measure the specific biomolecules that are actually secrete from the tears. So actually, the better we know our body, the better choices we make, right? The better decisions we make. So I hope this field would grow, and actually it's growing, and you can see that investments are going towards this field.
I think it was also affected by the fact, like, I don't know exactly, but on the COVID-19 outbreak, there was like demand for the tests, the one that is portable, the one that will be very easy to be manufactured as well.
Right.
So, was it PCR tests, like the fast PCR test that was putting like in the nose for the COVID-19?
Yeah, so we donate the samples from the nasal cavities, some people from the nasopharyngeal, right, and the throat. So we amplify the genetic material, right, and then we know. But we know that these viruses, they mutate quite fast, right, and there are different variants, and in such cases the sensitivity and specificity is the key, right, so that the primers that they have, they will be correctly actually diagnosing, so that there is no false negative result as a result, right. So antigen testing, so at home, right, antigen testing, lateral flow, right, so one would be dropping a sample, body fluid sample, and then observing and comparing to that of the control. But this is also based on the antibody-antigen interaction, right? Or in a research setting, we can also measure the antigen level using ELISA. Or, for example, also have our contribution to that as well. We have a paper published on that, the development of the biosensor, not biosensor, but optosensor. Optosensor is when we use aptamers as recognition elements while developing the sensor.
Instead of antibodies. So they're like antibodies, and aptamers are the one that is like detectable.
Yeah, so
Yes.
they are recognizing the target.
So basically, aptasensors are like sub part of biosensors.
Yeah.
Biosensors is the general name for it.
Yeah, so they would be functionalized over the transducer, and then while capturing the target, right, it produces the signal, right, changing the signal. So we're trying to see while there is no, for example, target, or there are some other targets they will not recognize, right, compared to that of when the sample or the aptamers have captured the antigen.
The thing that I just wanted to say, like the information about the COVID-19 is like in further 10, 20 years, if there will be such challenge that humanity will face, well, I guess the progress will go much faster because there will be a high demand for it. Probably.
Yeah, and plus, like for example, if someone is sick, right, and with contagious infection, nowadays we hear this news with mpox, right, monkeypox. We hear the news that about the Marburg virus, right, so that is being actually not circulated only in endemic areas, but also in some other areas of the world. So in such cases, we don't want to be exposed to those people, right? So having these portable detection tools allows at least to screen the patients, right? In diagnostics, it's always good to have two tests confirming the same results, because relying solely on one output would be quite, let's say, wise. So if we are, for example, rapidly detecting with biosensor the presence of this antigen, it's better to confirm it also bacteriologically if we're talking about bacteria, right? So that kind of reassuring that we're not having these false positive or false negative results. But it is one of the ways how to contain the infection, right, and how to limit the spread, right, and that's something how to end, for example, tuberculosis. If, for example, we know that this patient is suffering, obviously we don't want him or her to go to the diagnostic lab, right? By the time they would get, there would be a number of people, right, exposed to this pathogen. So in such cases, sensors, the rapid diagnostic tools could be of help.
Yeah, so hopefully in the future, when we'll be able to detect tuberculosis faster and more accurately using the biosensors as well, what kind of impact do you think will it have on the public health globally?
Well, globally, so let's take a look and a step back. So from the historical point of view, so if we look at the 18th, 19th century, you mentioned, right, so the tuberculosis was flourishing, but nowadays it's communicable diseases were flourishing, among them tuberculosis. But nowadays we have non-communicable diseases, right, metabolic disorders, right, which have increased drastically. So which means that we have learned how to contain, right, we have developed those preventative measures, the vaccine shots, but also we have developed the antimicrobial drugs, right, and that led us to get these superbugs, right, that are not susceptible to those antimicrobial drugs that we are using, right, to effectively kill them, but they have this resistance, right, in their genetic material to avoid this. So I believe that if we have enough tools developed that allow us to evaluate from different angles rapidly, specifically and sensitively, that at one point and we can reach, right, this goal that WHO set for us to eradicate tuberculosis by 2030. But before 2030 we only have about 5 years only left, right? So that is actually a challenge that we have. I would say one of the infections that has been eradicated is smallpox. So, and the next on the line we have tuberculosis. So, fingers crossed, and let's work together on actually tackling this issue.
Well, for sure.
Yeah.
If we are coming to the conclusion, well, I think you can start with the conclusion.
So, actually, I think that was a wonderful interview. I would really want to thank you for this very interesting insights on the tuberculosis, on the biosensors, on the aptamers, on how actually we are as community, as society, as humanity overall are heading towards this eradication of such a horrible disease as tuberculosis, and hopefully in the years future we'll be able to move on to finally finish off this tuberculosis epidemic, the disease, and move on to the next, to the treatment of other diseases which are on our list of
So for me personally, I'm an undergrad student. So of course, like in comparison with my fellow colleague, I have less. Obviously, I have less knowledge in biology, especially in microbiology sphere. Probably because I'm only second year medical student, but getting those insights, and I think we elaborated on the interview both on the topics that we are studying like very recently on Bio 120 courses. We only discovered PCR, and then on next year I'm personally like stepping in on the topic of aptamers, antigens, and then discussing about like this is the new feature that is going to be like replace these methods that will be more convenient. Like doing the new things is very like keeping me motivated as well to stay in this sphere. Probably to explore microbiology as well, but more from the clinical side, because I'm a medical student probably. For me, this interview gives a lot of new information, and I'm really now keen on biology, as I always am, because I basically chose this sphere by myself.
Yeah, thank you so much. But I want to make one note. We're not trying to replace anyone, but we're trying to complement, right, and to give the community like a choice, right, to choose like what kind of detection tools they want to use, right? Because depending on the sample that we analyze, I'm going back to biology, right? Some like techniques they could be more sensitive, right, or more specific. But then in that case, they have some other choices, and it could be also the aptamers that we're developing, right? So I'm trying to also contribute to this field, as most of our colleagues. We're not trying to replace anyone, because replacement is, I think, it's about the demand, right?
Yeah.
If the community doesn't have like interest, or they know that it is not sensitive or specific, or it's not working, right, or it shows the false positive or false negative results, then there's no demand, right? So in such cases, it just on its own will disappear, or may have like a lesser impact. So from my view, I'm not trying to.
Yeah, I think that biologists, as biologists, we should not like compete with each other, but rather work on a global goal. For instance, like treating tuberculosis, and here we should not try to come on top of each other and trying to like.
So thank you so much. I was really happy to actually be invited and to mentor you guys, and I really wish success to SciPod podcast, to have more like interesting speakers in the future. And you guys are wonderful, and you are doing a wonderful job contributing to the field of science, and specifically in Kazakhstan. I think it's something that we need to explore more, because we have great scientists, researchers in different fields, not only in biology, but yeah. So I'm wishing you best of luck, and I will show all my support if needed. Thank you.
Thank you.
Thank you very much.